The broadcast is now starting. All attendees are in listen-only mode.
Good morning and good afternoon to others. We'd like to welcome and thank you for attending our webinar on regulatory considerations and revalidation tests for managing change in packaging materials or critical supplies today. The purpose of this webinar is to provide you with the knowledge when a critical supply for your product or process changes. What are the regulatory items that you should consider? Regulatory bodies such as the FDA expect you will be able to do your due diligence in assessing the risk and impact to your product or processes. And in particular today, we're focusing on a change in packaging materials. And here to present are our resident experts, Sherry Robbins and Wendy Mock. Just for some housekeeping items, in the future, you can always go back and reference any of our webinars on our website. That's www.nelsonlabs.com. And they are under the Knowledge Center and the webinars page. They are usually posted about a week after the event. As for today, we welcome all your questions. You should be able to, you can submit them at any time, and we are reserving the last 10 to 15 minutes of the event today for Sherry and Wendy to answer as many as they possibly can. Just for a little bit of information and background on our presenters today, we have Sherry Robbins, who has worked at Nelson Laboratories for almost 14 years. She received her bachelor's degree in microbiology from Weber State University. Her current role is Director of Regulatory Affairs, where she hosts the FDA inspections and is responsible for overall compliance with the company. She has obtained a Quality Auditor and Quality Engineer certification through the American Society for Quality and a Specialist Microbiologist certification through the American Society of Microbiology. Wendy Mock received her Bachelor of Science degree in Biology from the University of Minnesota. She continued her education by obtaining an associate degree in clinical research. She has been with Nelson Labs for 7 years as the packaging section leader. She is a certified quality auditor and has obtained her registered microbiologist status with the American Association of Microbiologists. Wendy is actively participating in both the AAMI and the AFPM working groups. She was also published in the Pharmaceutical Formulation and Quality magazine. We'd now like to turn the time over to them to start our presentation.
Welcome, everyone. The first slide is on regulatory change cycle. First, we're going to start with this overview of the regulatory change cycle, because this slide represents a circle model for a reason. The point here is to close the loop. In this regulatory consideration segment, we'll start with the basic change management concepts, cover change awareness and detection mechanisms, discuss how to assess risk, how to mitigate those risks, and how to close the loop with implementation. Change management. The objective of a change management concept is to manage the change, not be managed by the change. The deliverables of a robust change management process will result in maintaining a validated state for your processes and products. Not only is this change management a regulatory requirement, but it can be used as a critical business tool to control your supply chain. Using a change management system to capture and house all critical supply changes will provide you with a repository for your regulatory documentation. It provides a methodical approach in showing your due diligence in managing product or process risk to regulatory agencies. You must be inspection ready at any time during the change process. So people in your organization need to fully understand the why behind the change management process in order for this to be successful. So with that said, I urge you to rely on a robust change management process and culture to accomplish this effort. Awareness and detection. In order to trigger the change management process, you need certain mechanisms in place to make you aware of the change. Notification mechanisms such as change agreements or notification lists are helpful, but they are not 100% guaranteed to be reliable. Some manufacturers may predetermine that the change that they make is not consequential enough to trigger notification. So as a company, you need to inspect what you expect through detection mechanisms. Regulatory authorities will hold you as the manufacturer responsible for any changes that your suppliers make that could affect your product or process, even if you were notified or not. The supply chain control has been a very hot topic with the FDA over the last few years. Risk assessment. The next step is to identify any risks that this change may pose to your product or process. The more stakeholders you involve, the more comprehensive this evaluation will become. This is exactly what you want. Take the time to include experts from your quality department, your engineers, purchasing, regulatory, maybe even your testing lab in order to cover all of your bases. The scope of your risk assessment may also differ based on the intended use of the product. For example, let's say your packaging supplier changes the product and you use the supply for two different product lines. One is sterile and one is not. What do you need to assess for each one? The risks to the process and the mitigation steps you will need are very different because of that intended use. But in addition to just regulatory risks, you should also be analyzing other types of risks, such as business or customer impact. For example, a change to supply may result in changes to your model or availability. So think about how that can affect your customer or client. Finally, tabulate your risk scores and justify the rationale behind that score. This will allow you to prioritize your mitigation efforts. Mitigation. The goal of mitigation is to achieve an acceptable level of risk. Start this process by putting together a mitigation strategy for your highest risk scores. If there is an adverse impact identified by the risk assessment, it needs to be mitigated down to an acceptable level. So determine what is an acceptable level for the intended use. Does it really have to be 0? Include your rationale as to why the risk threshold is acceptable. That way, the thought process is justified and documented, and it's not left to future interpretation. Next, decide what your mitigation actions will be. These may include acceptance verification testing by bringing in new sample lots to ensure there is no impact to the product or process. Maybe you will determine that the changes are too extensive and an entire revalidation is warranted. Or it is possible that equivalent be divided by the supplier and included in your risk rationale. For your mitigation planning, discuss who will do what are the target dates for completion, and what evidence is needed for the file. Implementation and dissemination. Finally, the last step is to close the open loops from the mitigation activities. Ensure that the actions taken are appropriate, adequate, and sustainable so that the risks identified are no longer an issue. Ensure that the change management is disseminated to all parties who need to be aware of the change so documents can be updated as needed. For example, purchase orders, procedures, supply specifications, regulatory submissions, and in-house regulatory files are all things you should consider in updating those documents. Ultimately, the objective of this whole exercise is to be able to demonstrate to regulatory authorities that due diligence has been performed. Awareness of the change is apparent. The impact and risk to the product or process has been assessed. The risks are mitigated with an established plan to address the risks. The impact has been addressed, and the loops are closed on implementation and documentation. Now, this concludes the regulatory consideration of the webinar. If you have determined that a change in packaging warrants revalidation or verification efforts, the next segment will cover how to approve that task. So we'll to Wendy to take it away.
Thanks, Sherry. Okay, revalidation. Sherry's example was a packaging supplier change. That falls into a sterile barrier system change. Other reasons that you would need to revalidate include process equipment, a change in device, or even a change in sterilization. Additional thoughts: what if you take your transportation system from a local delivery to a larger scope? Those type of things lead to a revalidation. And if you decide that revalidation is necessary, you need to take a look at what type of revalidation are you going to use. Are you going to do a complete revalidation, or are you going to look at an equivalency study? The key point with the equivalency study is to replicate what you did in the original validation by following the same tests. So when you're looking at revalidation, you need to make sure that you're aware of changes that are happening. Some changes can be so minor that as you're going through the process, you decide that it's not necessary. Just go ahead and record what you're looking for, and you can continue to do that. The next item is if it's a periodical review. And when you're doing this, look at if you're doing like a sterilization. Take that opportunity to look at your packaging and see if any of those changes happen to implement or affect that method and document it. So any of those efforts in response to the supply changes should be based on the intended use of the product. So if you decide to go ahead and revalidate, you're going to need to understand the standards that regulate what tests to do and how to perform them. These are run by the AMI/ANSI ISO 11607, Part 1 and Part 2. Part 1 is the materials and sterile barrier requirements. This is going to talk about how to do the tests and what the requirements are. Part 2 is and going to be more for the manufacturing of those packages. It's important to point out that this standard is the FDA consensus standard. It is the easiest path to getting approval of your testing. The second document that I have listed here is TIR 22. This is a guidance document for the 11607 standard. You can use it to help explain how to carry out what the requirements are. So now that you have your standards, let's take a look at what do you do with your devices. What is a worst case? Because you're not just going to send nominal packages through the process. You need to look at worst case. And there are two areas. There's the device and the material. So the standard states that for a device, when similar medical devices use the same packaging system, a rationale for establishing similarities and identifying the worst case shall be documented. And what this means is to pick the device that applies the most stress to the package. Is it the bulkiest? Is it the heaviest? Those are some of the ideas behind establishing a worst case device. When you're looking at material, it includes whether it goes through different temperatures, pressure, dwell, or sterilization cycles. The performance testing shall be conducted on the worst case sterile barrier system, produce that process limits of forming and sealing, and after exposure to all specified sterilization processes. So you're going to take a worst case device, which is going to be either the bulkiest or heaviest, and then you're going to run it through a possible sealer with the different dwells and pressures, and then you're going to expose it to multiple cycles. So once you've established your device, you need to start looking at the packaging tests. If you're, like I mentioned earlier, if you're doing equivalency, you'll want to repeat the same tests that you did initially. If you're looking at a revalidation, you have a choice, and here's a chart with the different choices that you have. 3 categories you need to address for each, and that's the strength, integrity, and microbial barrier. Underneath each, you have a choice of tests. I'm going to go through these tests and tell you why you would want to pick one based on your samples. So let's start with the test. This is a seal peel test. It's a strength test. It determines the strength of the seal at a specific place on the package. Normal process is to use 2 seals. You'll use one from the manufacturers and then one from the user. The reason that we choose this is the manufacturers have normally already got approval for their device or their package, and they've set a spec. So if you use the manufacturer's spec as your acceptance criteria as being equal to or greater than, you know that you'll be successful. You can also use the seal strength test to help set your IQ parameters for your sealers. The way that the process works is you measure a 1-inch segment of the package along one of the seals, whether that's the manufacturer's or yours, and you cut the package so that there's approximately 4 inches of material on the side of the seal. We take this and we put the seals in an instron or tensile tester, and we pull the sample apart. The results are reported as load at yield, energy to break, and they'll give you a value. This is a quantitative value. The burst test is the next strength test. It's performed by pressurizing a package until it bursts. The test system works by presetting the pressure, and then we inflate the package until it bursts. The results are usually PSI. The reason that you perform this test is if you take your packages up into an airplane or you go through different elevations, they'll be subjected to altitude changes or pressure changes. This will tell you how your seals will handle those differences. Also, if you're using EO sterilization, those have different pressure vacuums and can put stress on the seals also. So the burst test is for those types of scenarios. The next test is a creep test. This is also useful where you're looking at the pressure over time. So we inflate the package to 80% of the burst test value, and then we let it sit for approximately 90 seconds. And what we're doing is we're looking to make sure that that package's seals don't creep to a fail. So again, EO, sterilization, and then air shipping are a byproduct of this test. Okay, we're going to talk about integrity tests. The easiest integrity test that you can do is visual inspection, and it's regulated by ASTM F1886. And the process identifies looking for irregularities in the sterile barrier system. You're looking for tears, holes, things like that. You're also looking for the presence of foreign material. You can also include dimensional accuracy. If you're running 1/4-inch seals, this is a great opportunity for you to make sure that you're only getting 1/4-inch seals or minimum. Frequently, we've seen samples submitted that the seals have actually shifted to the side, and they're definitely less than the required minimum. Seal integrity, are they homogeneous? Are they complete? And then if your samples go through steam sterilization, is there still the presence of humidity or moisture inside? Did any of the labels stain? So those are the things that you'd look for visual inspection. Our tip here is always, if you find that you have some defects, to keep those or take really great photographs and incorporate them into your training system. It's a lot easier to teach someone what a crease through a seal is by having a visual example rather than just writing it down. So that's a tip for today. The next test we're going to look at is dye migration. Dye migration is the process of injecting dye into the package. We allow the weight of the dye to sit along the seal for a specific amount of time. You have to be careful with this test because the dye can have a wicking effect through porous material. So if you leave the solution in the package too long, you could pick up false failures because the solution actually wicks through the package rather than actually coming through the channel. This is a test for seal failure. It is not a whole package test. The next option is dye immersion. This also uses a blue dye, and it is a test for rigid containers. If you have pharmaceutical products or syringes, this is an option for you. And the way that it works is we place the samples into a blue dye, and we put it under a vacuum for a specific amount of time. After that time's elapsed, we release the vacuum, and with the release, if there are leaks in the product, it will dry the dye into the sample. Now you can test those samples by either UV-Vis, you can do a visual inspection. Normally we would do visual inspection for just R&D purposes, and then for routine production we would go with the UV-Vis because it's a lot more sensitive. One of the key notes to performing this test is to make sure that the contents of your product are not inhibiting the dye. So making sure that when the dye comes in contact with your product that it doesn't change the dye to create a false negative. An example of this would be tartaric acid. It turns the dye clear so it looks like you actually passed the test. So if you have product that are affecting the dye, this is not an appropriate test. The next option is a whole package test. It is the bubble emission. The way that it works is you inflate the package under a surfactant fluid, and you're looking for holes in a continuous stream. It's a very easy test. You have to be very careful because you don't want to put the pressure in the package to the point where you'll pass what's called a bubble point, and it looks like you're generating bubbles from all over the package. It looks like it fails. So making sure that you have the correct pressure for the inside of the package is a key to the test. Also, if you do this test on Tyvek materials and you notice that there is a slow leak or a slow amount of bubbles coming from the seal area, it's always a good idea to follow up Tyvek tests with the dye migration test. Tyvek sometimes has the capability of delaminating inside the material, but that doesn't affect the integrity of the package. So by following up a bubble emission failure of slow leaks with a dye migration test, you can confirm that it is a true failure rather than just the Tyvek delaminating. The next test is the microbial barrier tests. The first one we're going to talk about is the FDA consensus standard. It is AM-STM F1608. And this is a test for porous material. So if you have foils or films, this test is not appropriate. The way that it works is you take a 47 millimeter disc and you place it into a chamber. We expose it to Bacillus atrophius for 15 minutes. We then remove the filters, we blend them, and we plate them to determine the quantity of organisms that penetrated through the porous material. The fun part about this test is packages are supposed to maintain sterility, and so when we get counts on our plates, people start to get nervous because it looks like the package is not maintaining a sterile barrier. But it's an LR test, so it's going to give you a value, and this value actually is a ranking. And so the higher the ranking, the better the materials. So that's ASTM F1608. It's a material test. So if you have a rigid container for microbial, or if you have foil pouches, what do? Then this is an option for you. This is the Microbial Aerosol Challenge. It is a whole package test. And it works under the same theory as the F1608 test, such that instead of using a disc of the material, we're actually going to place the entire package into a chamber. And we're going to expose it again to the same organisms, except this time we're going to do it for a longer period. The F1608 test was only for 15 minutes. This one's going to actually go for 60 minutes. And that's going to give you a higher concentration of organisms in the chamber. So we expose it to 60 minutes of Bacillus atrophius, and then we evacuate the chamber, followed by letting the sample sit for a day. The reason that they sit is we're making sure that all the controls that are part of the test process are passed. After the controls are passed, we'll take those test packages and disinfect the outside. We'll move them to a clean area, clean room, and test the contents of the package for that indicator organism. This is a pass-fail test. The most important part to this test is to keep in mind the size of the chamber. It is a 36 by 36 inch chamber, which if you have very large packages, that is very difficult to do large quantities of samples. So making sure that you understand the size and the limitations is important when you test. Here's a picture of the indicator organism test. You can do the test either by immersion, we can do flush, or media fill. Flushing is for devices that are labeled sterile fluid pathway only. Immersion is taking the contents of the package and putting them directly into media. And then the media fills are for things that are like syringes that are pre-filled with media, or even ophthalmic bottles. So those type of things have been tested using this method. So those are the microbial tests that I'm going to talk about. We're going to talk about a little bit the rigors of transportation and then some accelerated aging. So transportation testing is managed with either ASTM or ISTA test procedures. The consensus standard is the D4169 test, and again, as I said earlier, it's the easiest path. The FDA has participated in these standards. And what they evaluate is the distribution, the handling, and the storage of the packages as they go through the system. So the distribution is going to be how they handle the shipping. The handling is how are they stored, and then the storage is temperatures. So the section of the standard, which is 6.3.5, states that the system shall provide protection to the package through all of these processes. So as you get into these standards, you'll identify different levels of tests, such as drop testing, vibration testing, stacking, climate pressures, things like that. So if you're interested in learning more about transportation tests, please feel free to give us a call. The accelerated aging is a way to get your samples to market faster. It's regulated by ASTM F1980, and the theory is to demonstrate that the package is not affected by the aging to the expected shelf life. It does not impact the performance of the package. The key to this test is to understand that it does not replace real-time aging. Real-time aging is the requirement. Accelerated aging is the option. So the test method is based on the observations of the Arrhenius equation. This is the equation, and it calculates the aging factor, which is what is the temperature that you want to use above ambient. So normally tests are aged at 55 degrees. Additionally, we are using a Q10 reaction doubling rate, which is normally a value of 2. I've only seen one person change this value, and it was a packaging engineer that was very smart and knew all about the thermal glass transition points of the plastics involved in their product, and was able to calculate a value of 1.8 for his Q10 versus the conservative value of 2. The gist of the equation is that 1 year of real-time aging is equivalent to 46 days using a value of 55 degrees Celsius for aging and an ambient temperature of 25 degrees Celsius. So when you're doing your accelerated aging, it's important to make sure that you justify your conditions and duration. The ambient temperature that FDA looks for is at 25 degrees. This is a value actually that's really very warm. Most warehouses are a lot more controlled than this. And if time is an issue, this is the value that you would want to look at possibly. The lower the ambient temperature, the faster your samples will age. And if you have documented evidence that your warehouse or the facility that your products are going to be located at are more than 25 degrees, go ahead and use that. Additionally, you want to make sure that you're making sure that your TG, which is your thermal gas transition point for polymers, is at least 10 to 15 degrees below that value. What happens if you approach it is once you put your samples into the aging chamber, they come out all melted, and that's not good for anybody. Additionally, you want to make sure that you consider relative humidity. The standard indicates that humidity is not a factor for accelerated aging. So when you use your laboratories to do accelerated aging, make sure that humidity is controlled. Humidity has no effect as part of the accelerated aging. If you want to look at humidity effects on packages, you should do that as part of the transportation storage tests, and then along with climactic stressing and that type of stuff. So don't do it as part of the accelerated aging. Question that I get is, should it be done with or without product? And that depends on what you're going to do with the packages. If you're going to just look at packages and evaluate them as part of a validation, then you don't need product in there. But if you're trying to do an evaluation from the beginning, if you put your product in there, it's almost like a 2-for-1. After the testing is done, they can return the products to you, and you can do functional analysis after that aging time period. If the package and product is expensive, you can always use stimulants or facsimile devices. Cosmetic defects are also acceptable. So I'm going to wrap this up. The summary is that if you're getting a supplier change, to make sure that you perform your risk assessment. Make sure you check off all those things that Sherry mentioned. You move forward with testing, define that testing, and justify why you're doing that testing. Perform the testing, and then document everything. And I think that wraps up this webinar. Mike has some information.
Great. Thank you, Sherry and Wendy, for your time and expertise today. We've had several questions coming in, and we are going to try and answer as many of those questions as we can. That'll be Sherry and Wendy answering respectively about regulatory and then also package testing questions as well. So I will turn the time back over to them. We'll take questions for about 10 to 15 minutes, and then we'll just wrap up a little early today. So I'll turn the time back over to them.
Okay, so a question we had was, what if you change cleaning and packaging suppliers, but you keep the packaging and sterilization dose the same? Can you justify not doing a new package validation? I think the key here is we have to determine if it's equivalent. It's all based on the intended use of your process. So if the packaging change can be demonstrated to be equivalent, then you have something to base your justification on for not doing a new package validation. The question is, how do you prove equivalency? Is that information that you have to prove out through testing to verify equivalency? Or is that something that you can get from maybe the manufacturer? Maybe they've done some equivalency testing for you. Okay, I think the next question we will turn over to Wendy.
Okay, so what is an acceptable LRV for microbial spore challenge testing? That's a very hard question. The way that I would approach it is I would find out what the values are that the manufacturer that you purchased the pouch from has established for their product and use that as a minimum. What I can tell you from experience is that normally Tyvek is about 5.2 or higher. Papers are usually around 1.5 to 1.8, and then when you start coating them, they move above the 2 values.
Okay, another question is, what about reassessment of product biocompatibility when packaging materials are changed? This again comes down to the intended use of your product. I think absolutely, and there are some quick and easy turnaround time, cheap cytotoxicity tests that you can do, you can perform that will help in that initial screening. And that will be able to detect if that causes any sort of cytotoxic effect to your product in that material. So I definitely think if that is a concern of the intended use of the product, that is something that could be easily checked off through a quick cytotoxicity test.
The next question that we have is, if we are using 2 sterile barrier systems, should we test the inner and the outer, or just one of them? Should we have 1 or 2 sterile barrier systems? The way that answer is approached is dependent upon where your labeling is. If your labeling is on the exterior of a double pouch system, you're claiming that the contents of that package are sterile. So you would need to look at both. If you claim that the inner pouch is only your sterile barrier, then the outer one suddenly becomes a dust protector. One note on that double pouch system, though, being the sterile barrier system, is that if you do all the testing on the outer pouch, I would recommend only doing minimal testing on the inner pouch, something like maybe a dye migration or a seal peel. Microbial is probably not necessary at that point because you have done it on the outer pouch, but maybe just an integrity and strength test to verify that it's doing what the purpose of it is for.
All right, the next question is, do you have any recommendations for routine monitoring of a validated lid stock tray, for example, daily burst testing? My recommendation here is it's based on the intended use again. What is the risk if something were to go wrong? And that will drive how diligent your efforts have to be in that routine monitoring. If the risk is low and that can be justified, maybe that's something you just sample on a routine basis every quarter. But if it's something that, if there's a change in that, have a very high risk impact to your product process, and you need to be more diligent in the inspection piece, then maybe that's every lot verification, and it's quarantined until you have some sort of assurance that there's no issue with that lot coming in.
So I got a couple of questions about the rationale for not including humidity as an important mechanism in accelerated aging. Humidity is, according to the standard, it's not supposed to have an effect on accelerated aging because we're supposed to be evaluating the packaging over time using a temperature. The humidity is not actually incorporated in any manner, shape, or form. But that being said, you still need to control the humidity, making sure that it doesn't have huge swings, because once you start incorporating swings into the aging process, it's no longer aging, it becomes climactic simulation. So making sure that that's controlled at a single point with its tolerance is important as part of the aging. You're not looking to evaluate humidity over time, you're looking to evaluate the product at a specific temperature.
All right, if you plan to purchase the same material from a new supplier, what are the tests that must or should be run to determine that the materials are indeed the same? And this is a great equivalency question, but there's more to just equivalency of the product here. If you're changing to a new supplier, there's more risk in that sometimes because you need to ensure that, their manufacturing process and their supply chain is strategically aligned with your regulatory needs. Making sure that they can deliver that consistent supply is key in that supply chain management. So the tests that should be run to determine if the materials are indeed the same, you could do side-by-side comparisons based on the rationale of the intended use. But any sort of those tests that Wendy has just described in the webinar, those are things that you could run just to compare and build some sort of confidence that you're not just taking anybody's word for that equivalency, but you have actual substantial data on file that you could show your regulatory body. Thanks.
Okay, I have a question regarding dye immersion tests. Is it suitable for closure systems composed of semi-rigid materials like blisters or aluminum foils? And the answer for that is no. Dye immersion testing uses a vacuum, and a lot of times foil pouches or blisters will pop as part of the vacuum test. So it's specifically only for rigid containers. The next question that I have is, when aging is performed without product, what is an acceptable percentage of degradation on the material that could be considered stable? That's the purpose of the aging, is to evaluate the package over time using the test methods. So determining an acceptable amount of degradation really isn't part of the testing that's designed around that. Usually what happens when you do the aging is you take the samples that are post-aged and run them through the dye migration test or the seal peel tests. What we've seen, if you're looking at seal peel tests, is that a lot of times the seal will actually get stronger over time rather than weaker. So those tests really aren't indicators of degradation. The next question is, we identified the FDA recommending 25 degrees as an ambient temperature. In which standard is this captured? It is in the ASTM F1980. The examples that they use is 25 degrees and 60 degrees. An important point to note that is, if you go outside of the recommended range, especially for the aging temperatures, the 55 to 60s, you'll definitely have to provide a significant amount of justification, because a lot of times the higher the temperature, the faster that the materials degrade or approach that thermal-gas transition point. Okay, we can do a couple more questions really quickly. Okay, on the slide for F2096, it was mentioned that the package is submerged into a surfactant solution. Is this unique to Nelson? The ASTM method only indicates to submerge underwater. I don't think it's unique to Nelson. My conversations with other ASTM committee meetings, several people use different types of surfactants. What it does is it increases the sensitivity of the test. So I don't think that this is a unique solution to our company. The next question, why do Microbial challenge if you have previously proved whole package integrity? ISO 11607 seems to require only one integrity test. Where and when would a case be where you would choose Microbial testing over F2096? So the standard calls out that you should do a Microbial and an integrity test. If you have data, you can use those to make a correlation between the two and then only do an integrity test. I don't know of anybody at this point that has made the case where they have integrity tests representing microbial testing, using the data for correlation. I'm just looking through here. The minimum seal requirement of a 1/4 inch is in ASTM F1886, or is this an industry? It's neither. I just used a 1/4 inch as an example for what to inspect for dimensional accuracy. Okay, we're going to do one more question and we're going to wrap it up. Okay, we are changing the material of our thermoform tray from PVC to rPET. This tray has a Tyvek lid and is also our master challenge device. We assume this needs to be revalidated. This is a significant change because you are changing your material. So revalidation would be required, but to what degree is dependent on your risk assessment, as Sherry pointed out. Making sure that you address those risks can either lead you down a complete revalidation or a shortened version for equivalency studies. So addressing which one you need is based on your risk assessment, which is a great way for us to summarize everything that we've talked about today. So thank you.
Okay, thank you everyone for attending today, and we'd like to thank Sherry and Wendy again for their time and sharing their expertise with us. This website, or this webinar, will be available approximately 1 week on the Nelson Labs website, so that you can review again the information there. And again, I'd like to have everyone have a great day, and keep looking for future webinars on our Nelson Labs website as well. Thank you.